The broadcast is now starting. All attendees are in listen-only mode.
Hi everyone, and thank you for joining us today. We've logged on to the Nelson Labs webinar, The Fight Against Viruses, the Importance of Cleanroom Virus Disinfection. Now, if you missed one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following on Twitter or LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There, you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and personal care industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now, let's get started. Today, we're joined by Dr. Candice Fox. Dr. Fox is a distinguished virologist and the department scientist in virology at the Nelson Labs Bozeman facility. She holds a PhD in biomedical sciences from the University of Central Florida, where she focused on a wide array of viruses, including SARS-CoV-2, Zika virus, and others. She has led significant research on infectious diseases and played a crucial role in developing novel antimicrobial nanoparticles. At Nelson Labs, Dr. Fox enhances our capabilities in virology, serving industries such as consumer testing, medical devices, and pharmaceuticals. Her extensive experience with BSL-3 laboratory techniques, project management, and the development of experimental assays makes her a pivotal member to our team. Dr. Fox's deep understanding of viral infection processes and replication assays contributes significantly to our mission of safeguarding global health. Dr. Fox, it's great to have you today. Now, we do encourage your questions throughout today's presentation. Submit them at any time in the questions box. Dr. Fox will try to answer as many as she can in the last 10 to 15 minutes of the webinar. And now, beginning today's presentation, I turn the time over to Dr. Fox.
Thank you for joining us today. Today in our webinar, we will be discussing cleanroom virus disinfection validation testing and the importance of these tests in our ever-present fight against viruses. Specifically, we will go over cGMP regulations, principles of virucidal disinfection, and how these disinfections are actually evaluated for their efficacy, and how validation programs can be implemented in your specific manufacturing facility. A great way to have an overview of the current good manufacturing practices, or cGMPs, are a principle called the 5 Ps. And this encompasses people, processes, products, premises, and equipment, and procedures. Ultimately, all of these concepts lead to ensuring the end product have the utmost highest quality whenever dealing with manufacturing these pharmaceuticals. So for example, the products must have specifications for raw materials, components, intermediate, and finished products. They must have repeatable methods for research, development, processing, manufacturing, and packing, testing, sampling, status control, and stability testing and record keeping. To ensure all these criteria are met, different safeguards are implemented, such as ensuring that the premises and equipment can actually handle the production of these products. So the premises and equipment need to be validated and calibrated for the expected performance. They must have procedures, schedules, and records associated with every procedure that is occurring at the particular manufacturing premise. And they need to have design for effective cleaning and disinfection. Another concept that is of utmost importance for premise and equipment development is to ensure it's designed to prevent cross-contamination with different products and different viral for example, contaminations. So within the current Good Manufacturing Practices, or cGMPs, the proper building design and cleaning procedures are actually the second listed right after personnel. And that suggests that this is a very important concept that needs to be implemented. So if we specifically look at the Code of Federal Regulations 21 Part 211, which is the current good manufacturing practice for finished product pharmaceuticals, we can look at the design and construction features highlighted in 211.42. And this specifies that any building or buildings used within the manufacturing, processing, packing, or holding of a drug product shall be of suitable size, construction, and location to facilitate cleaning, maintenance, and proper operations. And this includes aseptic processing, such as containing floors, walls, and ceilings of smooth, hard surfaces that are easily cleanable. And additionally have a system for cleaning and disinfecting the room and equipment to produce these aseptic conditions. So for example, here's pictured a typical clean room that can be utilized for pharmaceutical manufacturing. We also need to design these clean rooms to be able to be properly controlled of the microbiome contamination. So that needs to include appropriate written procedures designed to prevent any microorganisms in the drug products. And it needs to be established and properly followed throughout the entire facility. So these need to include appropriate written procedures designed to prevent contamination of drug products, and if they are being reported to be sterile, and they need to be established and followed. And such procedures should include the validation of all aseptic and sterilization processes. So if you look at this particular example of a clean room, you can see that there are no artwork, there's no accessories, there's no unnecessary equipment. What you do see is a space that is specifically designed to have readily cleanable floors, walls, and ceilings that have smooth and non-porous surfaces. So for example, you can see that there's the flooring that might be epoxy or laminate, for example. There's stainless steel tables. There's different pharmaceutical isolators that could be utilized. We have different rubber gloves, different pass-throughs that need to be, every surface needs to be specifically evaluated. And you could have glass that is present in the clean room. You need to consider the lighting and the different air filtration systems that occur within the clean room. And what is not present in the picture are actually any gloves that you might be utilizing to process the drug product. So for example, latex gloves. And then also if there are any carts that are utilized to carry equipment or supplies, including the wheels, you need to consider those surfaces as well. So in fact, the USP 1072 covering disinfectants and antiseptics, they actually have a table of typical surfaces to be decontaminated by disinfectants in a pharmaceutical manufacturing area. And it specifies, because a wide range of different materials of construction are typically used in clean rooms, each material needs to be evaluated separately to validate the efficacy of a given disinfectant. So for example, it lists different materials that are typically present. So you could have glass that are applied for the windows, for example, and also the vesicles for the specific drug products. Plastic, vinyl, curtains could be present. And so you need to properly validate every disinfectant that you're using on a surface within the clean room to ensure the efficacy of that specific disinfectant. And what happens when you do not follow the GMPs? Well, FDA conducts audits on sites, and they do so to protect consumers from unsafe products. And these audits can occur for various reasons, such as surveillance inspections, for cause inspections, application-based inspections, and inspections to verify the reliability, integrity, and compliance of clinical and non-clinical research. And when a site is found not to follow the cGMPs or their own written procedures, they may be issued an FDA Form 483, as shown here. So this is an example of Form 483 from the FDA from a presentation they did last year on inspections of drug manufacturing facilities. And with the redactions, you can still see that the site was adhering to their written procedures, but the problem is that the method of sanitation was not validated. And so for example, it lists here that the company did not validate the sanitation procedures employed, and it did not prove that the product quality was not impacted, and did not document if and which product drums were left in the room during fumigation, for example. So all of these proper procedures need to be well documented and well established in order to ensure your clean room is sterile. And so how can we test this? We can bridge the gap between GMPs and written procedures through validation. So Nelson Labs can help you with the validation of cleaning procedures. And disinfectants need to be validated because environmental contaminants can be introduced during any time during the manufacturing process. And effective cleaning and disinfecting procedures are critical for manufacturing facilities themselves. And the validation studies need to be tested against every surface type that is present. And additional concerns that need to be considered is that the disinfectant needs to be validated against known contaminants to ensure cleaning procedure is effective in clean and dirty conditions. So for example, there may be organic materials present on the surface, blood contact surfaces, and soil load present within the materials. So these all need to be considered when you're designing a disinfectant validation study. And how do disinfectants actually target viruses, and how do they work? So you need to understand these principles when choosing a suitable chemical disinfectant. And then effectiveness of a disinfectant depends on its intrinsic biocidal activity. So for example, these chemicals can target different components of viral structures. So for example, there are alcohol-based disinfectants that can target the virus lipid envelope. And then these are obviously also utilized as a general purpose disinfectant and an antiseptic. And then there are other chemicals that can target nucleic acids, for example, amino groups, and cause general oxidation. Disinfectant efficacy also is dependent on the concentration of the disinfectant. And so this should be considered when you are performing dilutions of your disinfectant. You need to ensure that the concentration you are actually utilizing to disinfect your surface is effective against viruses, the necessary contact time, the nature of the surface that is actually being disinfected, the hardness of the water used to dilute the disinfectant, and then the amount of organic materials present on the surface. An additional concern that you need to evaluate when designing a disinfectant validation study is the number and the type of microorganisms present. So for example, you could potentially have contaminating bacterial sources, so bacteria themselves, and then also spores, which are actually quite resistant to different disinfectants. You could also have fungal sources. And what we're going to be focused on today are viruses. So viruses can come in two different structures. They can be non-enveloped or enveloped. And these envelopes are typically composed of lipids from the host cell. And some examples of these envelope viruses include hepatitis B virus and HIV. And examples of non-envelope viruses are poliovirus and rhinovirus. And what is important to understand about the different structures of the viruses is that the physiochemical properties of these microorganisms can influence its sensitivity to disinfectants. So for example, envelope versus non-envelope viruses, typically non-envelope viruses are more resistant to disinfectant. So this table is depicting just that the different microorganisms can have different sensitivities to disinfectants. Up top, we have a list of different chemicals that are commonly used as disinfectants. And then the microorganisms are actually organized here based on their susceptibility to disinfectants. So we can have envelope viruses, non-envelope viruses, and then there are some specific viruses that are very hard to inactivate that are shown here. So as I mentioned, generally speaking, envelope viruses are more susceptible to disinfectant as compared to non-envelope viruses. So all these principles need to be considered when you are selecting a particular disinfectant to be utilized against a wide range of viruses. So in order to select a proper disinfectant, it's common practice to use multiple types and classifications of these disinfectant chemicals on a scheduled rotation. And the benefit of rotating through different chemicals with the different mechanism of activation can reduce the likelihood of microbial adaptation to a single agent, therefore maintaining the effectiveness of a disinfection procedure over time. So what this means is that if you continually utilize one particular disinfectant for the clean room, for example, then the particular microorganisms can develop and evolve counter actions to circumvent the ability of the disinfectant to inactivate it. And the regulations also recommend that the chosen disinfectants are registered with the EPA or the ECHA, so the European Chemical Agency. When we are particularly selecting a disinfectant for use in manufacturing for pharmaceuticals, there are additional concerns that need to be considered. So as we mentioned before, the number and type of microorganisms that need to be controlled need to be evaluated. The spectrum of activity of commercially available disinfectants, as I mentioned before, different disinfectants can have different mechanisms of action, and you should utilize multiple different modes of action. And you also need to understand if the claims as a sterilant or if it is registered within the federal regulation, so EPA or the European. You also need to consider the concentration, application method, and the contact time of the disinfectant, the nature of the surface material being disinfected, and its compatibility with the disinfectant. Another step of that note is that you need to understand the compatibility of this disinfectant with cleaning agents and other disinfectants and the planned disinfectant rotation in regards to that particular surface. So your cleaning procedure needs to ensure that the surface itself is not going to be harmed or degraded, basically, by your cleaning procedure. And that should also be addressed. So in order to determine the efficacy of a disinfectant within a pharmaceutical manufacturing setting, the following steps can be considered essential. So we can utilize use dilution tests, surface challenge tests, and statistical comparison. So the use dilution tests are basically tests that can screen disinfectants for their efficacy at various concentrations and contact times against a wide range of standard test organisms and environmental isolates. The surface challenge tests basically can utilize standard test microorganisms that are typical of the environmental isolates and then apply disinfectants to the surfaces at selected use concentration with a specified contact time, and then determine the log reduction of the challenge microorganisms. And then the third is the statistical comparison, and you can do this with monitoring and assessing the frequency of isolation and number of microorganisms isolated prior to and then after the implementation of a new disinfectant. So specific tests that can address the disinfectant's efficacy using the use dilution tests. These are basically for liquid test articles, so it could be hand sanitizer, liquid soap, and lotion. ASTM E1052, for example, is standard practice to assess the activity of microcidals against viruses in suspension. EN 14476 are chemical disinfectants and antiseptics doing a suspension test, and that evaluates virucidal activity in the medical area. Specifically looking at the surface challenge test, we can utilize these tests for disinfecting sprays, wipes, and solutions. And viruses are typically dried on the surface first for these challenges, and then the disinfectant is applied afterwards. So there's ASTM E1053 and EN 16777. An alternative approach is to test against antiviral treated or coated surfaces. So these are basically surfaces that are containing or coated by an agent that inhibits the ability of a microorganism to grow on the surface of that material. So they can cover antiviral treated plastics, paints, fixtures, medical devices, and textiles. And to go into specifics about the different methods that can be utilized for surface challenge methods are shown here. So we have ASTM E1053, which is typically utilized for a glass carrier, and you need to be able to recover a 10 to the 4.8 infectious unit per carrier needs to be recovered. And what I mean by that is viruses can be quantified based off of their infectious ability. So we can do TCID50 or plaque assays, for example, to measure this particular activity of the virus. And this method utilizes various viruses, and the acceptance criteria for this is there's a calculated log reduction of prior to testing versus after testing. And the methodology states that it should be effective against at least one non-envelope virus. The European standard, EN 16777, typically tests on stainless steel discs, where 10 to the 8 TCID50 per mil of different viruses, so for example, norovirus, adenovirus, and vaccinia virus are applied to these discs. And the acceptance criteria for this method is the 4 log reduction should be achieved once your disinfectant is applied. And then these particular ISOs shown here are relative to the antiviral treated surfaces. So ISO 21702 is for antiviral treated plastics and different hard surfaces like that, where 10 to the 7 PFU or TCID50 per mil of either influenza A or feline calicivirus, which is a surrogate for human norovirus, are applied to the treated surfaces. The acceptance criteria is that calculated antiviral activity is determined, and that can be reported to the federal regulations. ISO 18184 are for treated textile products, including fabrics, fibers, yarns, and braids. And in this procedure, 10 to the 7 PFU or TCID50 per mil of influenza A or feline calicivirus are applied to the treated textiles. And again, the acceptance criteria for this method is based on a calculated antiviral efficacy value to determine good or excellent effect. So how can these disinfectant validation programs be implemented, and where are some of the issues that come up when proper disinfection program are not evaluated or not developed? So basically, we need to ensure that written procedures are properly developed and that the staff performing the disinfectant are properly trained. Decisions on disinfectant rotation also need to be considered and properly documented. As I mentioned before, a strategy employing multiple different chemicals with different mechanisms of actions should be implemented. The institution of application methods and contact times need to be properly documented and implemented. Environmentally monitoring to determine the efficacy of the disinfection program is also critical. So you need to not only validate your disinfection program, but also ensure that there are no differences and new alterations to your procedure that could result in an additional contamination that you previously did not consider. The disinfection program also needs to consider personnel safety and ensure that the personnel and the staff that are actually performing the disinfectant are going to be properly protected from any harm or any safety concerns. Specifically, cGMP 21 CFR 211.67 details the equipment cleaning and maintenance, and it showcases the requirements for the written procedures for cleaning, maintenance, and sanitization of the pharmaceutical manufacturing equipment. And it details that these procedures should address the assignment of responsibility. So who is going to actually be responsible for the disinfectant of the clean room? Establishment of the schedules, so to ensure that the proper rotation of the disinfectant is going to be properly followed. Detailed of the cleaning operations, so specifically going into the contact time, the use, dilution, concentration of the disinfectant is very important, as we mentioned before. The area itself needs to be inspected for cleanliness immediately prior to use, and that the maintenance of cleaning and sanitation records also need to be properly documented and controlled. So why is an implementation of a disinfectant validation program particularly important, especially in regards to viral safety? So biopharmaceuticals are at a high risk for virus contamination, and this can lead to risk to patients, either by direct infection risk. So for example, the drug product is contaminated with a virus that can infect humans. That could lead to a cascade of awful risks. There's adverse reaction risk. So if there's something in the product that is causing reactions in the patient, there's reduced efficacy of the product. So then your drug product itself is not as effective against the target disease. And this can also lead to drug shortages. So if there is a contamination in the pharmaceutical product, and you're going to have to, shut down or, take back some lots, then obviously this will lead to drug shortage, and the patients are the ones that will suffer. There's a financial risk. Obviously, there's going to be investigations if there's a contamination in a particular drug lot, facility shutdown, and a market share loss. So for example, there was a well-known virus contamination in a biologic manufacturing in Massachusetts in 2009. And it was estimated that the manufacturing crisis resulted in $100 to $300 million loss in sales. So these really need to be properly prevented, not only from the health standpoint of patients, but also this financial risk to the biologic manufacturer. So regulations are in place to mitigate these risks. And Nelson Labs can help you meet your regulatory demands. In-house, we have over 50 cell lines, both human and animal. We have over 100 viruses, including avian influenza strains, SARS-CoV-2, rhinoviruses, and many more. We have BSL-2 and BSL-3 capabilities, and we have a robust surface coupon library that we can test against multiple disinfectants. And to highlight just again that the different structures of viruses can be employed by very different structures. For example, we have non-envelope viruses that can be DNA or RNA-based genomes. There are enveloped viruses as shown here. And you can see that there are differences with their size as well as their shape. And all of these different characteristics can influence the ability of the virus to be inactivated by select disinfectants. So today we went over cGMP regulations, how disinfectants can be utilized and target different components of virus structures, how these disinfectants are actually evaluated, and how the validation programs are implemented and issues that could arise if the validation programs are not properly implemented. Thank you for your time, and if there are any questions, please ask them now.
Fantastic. Dr. Fox, thank you so much for your insights. We've all learned some great information today, and you've given us a lot to think about. We did receive some questions as you spoke today that we would like you to address now. If we don't get to your specific question, our presenter will be happy to address it individually after we conclude today. And so for the first question, do you only select one virus strain per validation?
So it depends on the study, because you would need to consider the potential of contaminating viruses to test against. So you need to do a risk assessment for what viruses could be present in the environment. And in fact, regulations have recommendations for select criteria, and pairing with the proper testing partner can actually help determine the correct viruses to test against in your particular circumstance.
Fantastic, thank you. So the next question we have is, how often do you have to validate a disinfection program?
Validations should be considered when any new deviation is introduced in the manufacturing process. So for example, if say a new disinfectant is introduced, or if there are any new equipment or new instrument that needs to be disinfected, that should also be evaluated if your select disinfectant can work against that particular surface.
Fantastic, thank you. The other question that we received was, are there any examples of disinfectant validation protocols specific for viruses?
Let's go back to, so for example, this is a table comparing the different methodologies for testing disinfectant efficacy against viruses. So these are specific for on surfaces. There are also additional methods that are specific for viruses in suspension.
Fantastic, thank you. And then it's, can you test on multiple surfaces within a single study, or is it one surface, one study?
That's a great question. So the USPA, the USP states that you need to test against every material that is present in your clean room, and that these should be evaluated separately. So within one study, for example, you can have multiple surfaces be evaluated. However, you do need to ensure that your disinfection program is validated against every surface present. And here at Nelson Lab, we actually have a very robust surface coupons, a library that we can test against.
Fantastic, thank you. And then the last question that we'll have time for today is, can you run one method to cover multiple agencies? For example, European versus FDA.
Right, that's a good question. So we can modify our different testing procedure to comply with different tests that you can then utilize to apply to the regulations.
Fantastic. Thank you, Dr. Fox, for that thorough and informative presentation. Your insights are deeply appreciated, and we can now walk away with a deeper understanding of viral cleanroom disinfection than when we began today. And I'd like to thank you, the viewer, for attending this session. I hope that you found it to be a valuable experience. We appreciate your time and support in helping us safeguard global health. This now concludes today's webinar, and I hope you have a wonderful rest of your day.